this video we will discuss about this. We will continue our discussion about this FCPR03 that works similar to the way this PDE4 inhibition works, that is by reducing the ER stress. So in this figure, the panel on the left side, will considered by by inhibiting this ROS to see whether this if we can inhibit this ROS to other ways, that will give to the same result that FCPR03 shows. So in this, what they did is they first introduce this ER stress with the help of this oxygen glucose deprivation condition, and then because of this the ROS generation will increase within the cells, and then they add this NAC, that is free radical exchanger, so it will neutralize this ROS. So finally what will happen is that the ER stress should get reduced. So what they check this approach through the same experiment that they used in the previous studies, that is they did the Western blot to check the amount of proteins that are involved in ER stress are affected or not, and they find that when both the ER stress and this NAC, this free radical exchanger, is added, we can see that amount of ER stress, amount of proteins that are involved in ER stress is reduced, as we can see in GRP78. That is, when the quantitative analysis is done, it can be seen that it is significantly reduced, and same is done with PDE4 alpha. We can see that compared to the third case where there is no NAC, we can see that in phosphorylated EAF2 alpha, the amount is reduced in the case where both the ER stress and free radical exchanger is there, and same is been shown by quantitative, semi-quantitative analysis of this Western blot. And this is the fluorescence of the experiment. We can see that first one is the controlled experiment, and the bottom one is the ER stress experiment. We can see that there is a and in this cells are stained, the ROS are stained through this red dye. We can see that when the FCPR03 is there is the decrease in the amount of this fluorescence, and similar kind of decrease is seen when the cells are treated with this free radical exchanger, that is NAC, and both of them, the signals in both of them are reduced compared to the case where there is only ER stress. This thing is quantified, this intracellular ROS is quantified through this image analysis of the confocal image analysis of the confocal microscopy image, and we can see that the reduce in the amount of ROS is similar in the case where cells are pre-treated with FCPR03, and cells that have this free radical exchanger, that is NAC. Later on they also check the MDA level, or we can say the ER stress level through this MDA, and they can see that there is a decrease in the amount of ER stress, and it is similar in both the case where cells are pre-treated with FCPR03 or cells that have this NAC. And the cell viability, in the cell viability assay, we can see that the cells that are treated, the cell viability assay, we can see that cells that are treated with only with ER stress has very low viability, and when the cells are pre-treated with FCPR03 or oxygen glucose deprivation, their cells viability increase slightly because they might reduce the ER stress by neutralizing the ROS. or oxygen glucose, or neutralizing the ROS by through this free radical scavenger, or uh, in case of NAC, or we can say by increasing the amount of HO1 or hemoglucosinase 1 in case of pretreatment with FCPR03. The panel on the right figure represents panel on the right figure functions by inhibiting works on the function of inhibiting this NRF2. And if you need to understand the overall picture, you can refer to the previous video. So NRF2 will uh, move into the, so for a brief description before we move in this panel is that NRF2 will move into the nucleus that will trigger or that will upregulate the production of HO1. So in this panel what they have done is that they have inhibited NRF1 and check whether this will also results in the this will also results in the inactivation. This will result in the inactivation of FCPR03 or not. So this will make sure that FCPR03 only works through this way and not through other way to reduce the ER stress. So they, in this figure, they have identified they have checked the protein, that is PEIF2 alpha, and they can see that there is slightly reduced compared to the case on the third case where there is no FCPR03. And when they did the semi-quantitative analysis, they can see that cells they can see that the phosphorylated EIF2 alpha is reduced. Phosphorylated EIF alpha is reduced in the case when the cells are treated with both oxygen glucose deprivation and FCPR03. So in this case they have not inhibited NRF2. That's why these results are similar to the results that we have seen in our previous videos. Here they have inhibited the NRF2 in this figure, panel E, and we can see that cells that are treated with ML385, it will inhibit NRF2. Cells that are treated with ML385, this the cell viability is reduced compared to the case where cells are not treated with ML385. So this indicates that NRF2 could be the major pathway by which FCPR03 works to inhibit ER stress. And this is the flow cytometer assay. In this we can see that in the control, compared to the control figure, when there is ER stress, there is more NRF2. In this figure, NRF2 is stained with the red dye, and when the cells are treated with pre-treated with FCPR03 and there is no ER stress, it looks like this. And when the cells are pre-treated with FCPR03 and there is a ER stress, then the signal is more than this, more than the top figure, but it is lower than the left figure. So this shows that FCPR03 helps in the reduction of the ER stress, and both these fluorescence figures are quantified. The fluorescence figures are quantified in this panel D. We can see that the cells that have that are pre-treated with FCPR03 has more cells that are pre-treated with FCPR03 and oxygen glucose deprivation has a high number of ROS, but it is lower than the case in which cells are not treated with FCPR03, and it is more than the case where cells does not have ER stress. So this thing depicts the quantitative analysis of this figure. I hope you have understood these two approaches that are used to validate the point that FCPR03 works through this NRF2 works majorly through NRF2 pathway to reduce the ER stress, and thus helps in blocking neural damages. If you need to understand this thing or this story in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.